Hello everyone. With this video, let us start with a study of the Zoology 4th semester paper, which is introduced under NEP scheme. The title of the paper is Gene Technology, Immunology and Computational Biology. So I'm going to start with the first unit, which covers principles of gene manipulation. So this is the first session uh, in which we are going to study the recombinant DNA technology. What is recombinant DNA technology? Recombinant DNA technology is a method of manipulating DNA molecule. You know, DNA molecule is the genetic material which consists of genes which control the characters of the organism. So you wish to introduce character of one organism into another organism. So there we are going to use this recombinant DNA technology. So basically, recombinant DNA technology is a method where you are going to cut isolate DNA from one organism, cut the desired part, and then introduce that desired gene into the target organism. And this technology is a very important technology used in genetic engineering, and it involves many enzymes and techniques, etc. So pictorially, you can see here, this is a chromosome which consists of the condensed DNA molecule, and DNA is a double helix molecule which consists of genes. So different genes which are producing different kinds of proteins, and they control different characters. Here, in brief, you can understand what is how the biotechnology, how the recombinant DNA technology works. This is the cell of our interest organism, and I want to isolate the gene from this particular organism, right? And the gene of interest, to isolate the gene, first you have to isolate the DNA of the uh, organism, and the isolated DNA, you are going to cut the desired gene, and like this I'm going to isolate the gene of my interest, right? So to isolate the gene you have to cut the DNA, and here we use many type of enzymes, especially the endonuclease enzymes. And this gene of interest is going to be introduced into a vehicle, because most of the time you cannot directly transfer it to the target organism cell. So you need a vehicle, which is commonly called vector. And there are different kinds of vectors which can be used in recombinant DNA technology. In this particular example, they have shown the plasmid, which is obtained from the bacteria. So you are going to take the bacterial plasmid, which can be used as a vector. You cut it with the same enzyme, and you can insert the gene of interest into this vector. And now you can see here, this is a plasmid DNA, and now we have inserted the gene of interest into this plasmid. So this is the recombinant DNA, because it is a combination of two different DNAs. So we are going to introduce this recombinant DNA into the target cell, and once it gets inside the cell, so there are different methods of introducing this into the target cell, and once it gets inside the target cell, it starts producing the gene of our interest. Remember, this gene which we have introduced, it is foreign to this particular cell, but because of the presence of this gene, it starts producing the product which is related to this gene. So like this, in the recombinant DNA technology, you can introduce gene of one organism into another organism. So recombinant DNA technology involves a series of steps to create a new DNA molecule by combining segments of DNA from different sources. The basic steps involved in recombinant DNA technology are as follows. Isolation of DNA. So as I already mentioned, this is the first step. So once you identify the gene of your interest, First, you need to isolate the DNA from that uh, organism cells. So by different techniques, you can isolate the DNA. Once the DNA is isolated, you are going to cut the gene of interest. So only the specific genes I want to cut. Therefore, we are going to use the specific the enzymes which are going to cut the required gene only. So endonucleases are the enzymes which are used for this purpose. Once the gene of interest is cut from the DNA, and the next step is creation of recombinant DNA. So this is where we are going to use the vectors or phages, etc. So using different techniques, you are going to create a recombinant DNA. So what is recombinant DNA? It is the DNA of two different organisms. So you are going to join them using enzymes like ligase. So once the recombinant DNA is created, you are going to introduce that into the target cell, the organism uh, of your interest, where you want to express that particular gene. So that is introduced, and there are different techniques for introduction of recombinant DNA into the target cell, like electroporation, transformation, etc., which we will study in another topic. And then once you introduce the recombinant DNA into the target cell, uh, all the cells, all the target cells may have not taken that recombinant DNA. So you have to select the cells which have taken the recombinant DNA. So this is an important process where you are going to select only those target cells, which have taken up the recombinant DNA. And once you identified the cells with recombinant DNA, you are going to use them for whatever your requirement. So that is a characterization of the recombinant DNA. So pictorially, here the same is depicted. So this is a bacterial plasmid, and this is a site. Different sites are there, which we'll study later. We are going to cut the gene from the source and the plasmid. Remember, to cut the plasmid and the gene, the same type of endonuclease enzyme is used. So it is cut here, and the gene of interest is inserted into the plasmid using ligase enzymes. Now this is the recombinant DNA, and this recombinant DNA is introduced into the bacterial cell using different methods. You can see here, this is the bacterial DNA, and this is the DNA, recombinant DNA, which is introduced. And this bacteria is cultured in a bacterial medium, and the colony which is formed. So we are going to select only the cells which contain the gene of our interest, that is the recombinant DNA. And after identification, so we are going to use it for the further processing. So here is another example, like the gene is obtained from Agrobacterium tumefaciens. T4 plasmid is here is tDNA, that is Ti plasmid is used here as a vector, and the gene of interest is introduced into a plant cell, right? So, for example, you may be familiar with golden rice, where the beta carotene gene from one organism is introduced into the rice plant, which produces the golden colored rice, that is with beta carotene protein. So, like this, you can introduce a gene from one organism into a plant, so where the plant starts producing a new product. Applications of recombinant DNA technology. Recombination DNA technology is a very important gene manipulation technique, the technique in genetic engineering, and it has a very wide use. Important uses in the field of medicine, like production of hormones, proteins, enzymes, vitamins, etc. And in agriculture, you can use this technique to produce disease-resistant crops and crops with desired characters. And in the genetic research, the recombinant DNA technology can be used to study the genes, isolate the genes, understand the genes, etc. For example, therapeutic products. For the production of therapeutic products like vaccines, hormones, antibodies, recombinant proteins, anti-cancer drugs, and many other products which are very important for the medical treatment. For example, the creation of vaccines against viruses, deadly viruses, etc. So they can be produced very accurately, they can be produced in a very short time using recombinant DNA technology. And to produce genetically modified products like GM fruits, vegetables, crops, genetically modified animals. So here we are going to produce the fruits with some desirable characters from other organisms. So nowadays, genetically modified fruits, vegetable crops, and animals have become very common, and they are available in the market. And in the diagnosis and treatment, like gene therapy, so which is going to be the future of medicine. So recombinant DNA technology is used, and in the creation of monitoring devices, health monitoring devices, they are going to use the genetic engineering technology. And when it comes to the environment, the DNA manipulation technique or recombinant DNA technology can be used to produce biofuels like biohydrogen, bioethanol, biomethanol, biobutanol, etc., which will replace the traditional fossil fuels, and that will be a good thing for the environment. And in the research, okay, for the research, you can use the recombinant DNA technology for isolation of gene of the interest, sequencing and studying the genes, identifying the gene function, gene silencing. So once the gene of our interest is identified and isolated and sequenced, so it can be used for many different purposes in the research field. So that's about the first chapter, and these are the references which you can use for detailed study. Thank you.